Hi, everybody, and welcome to the FRAXA seminar series. I'm Dr. Mike Tranfaglia, and I'm the Medical Director and Chief Scientific Officer of FRAXA. And next to me is my lovely wife and the Executive Director of FRAXA, Katie Clapp. As most of you have probably noticed, we've been facing some restrictions these days, and so we haven't had a chance to have in-person meetings this year. To try to make up for that, FRAXA is organizing a series of webinars to try to get the word out about Fragile X research. And most recently, we've been featuring some of the Fragile X Centers of Excellence. And one of the most excellent of all is the subject of today's webinar. Let me say a little bit about the format for this webinar. We'll have 4 speakers today. So each of them will speak in turn, and we'll save questions for the end. And if you haven't participated in these webinars before, the way you ask a question is through the chat function on Zoom. And so you can get a question in the queue at any point along the way, but we'll actually answer the questions at the end of the meeting. So to kick things off, I'd like to introduce Dr. Craig Erickson. He's a professor of psychiatry at Cincinnati Children's Hospital and the University of Cincinnati College of Medicine. Craig is one of the few other psychiatrists in the field, so it's especially good to have him with us today. He's going to talk about translational medicine and mechanistic studies of brain neurophysiology and Fragile X, and give us an overview of the NIH Center, for which he's one of the PIs. Craig?
Mike and Katie, thank you very much. And I, anytime I'm involved with the FRAXA event, it always reminds me that FRAXA was really responsible for my entry into Fragile X research about 15 years ago. The first grant I ever received was from FRAXA, and we've do what we do, and we've grown what we've grown in Cincinnati. Really due to FRAXA support that continues to this day. So really forever indebted to FRAXA and the donors and all of the forward thinking from your organization. And now I have the great opportunity to lead off and present on our NIH funded U54 Fragile X Center that I direct with Kim Huber at Texas Southwestern. And after I talk, Ernest Pedapati is going to talk about human EEG, Devin Binder about electrophysiology and studies in mice, and then Kim Huber is going to talk about her work in the slice and at more of a microscopic neuronal level. So really excited to do this, and I'll kick off our overview here. Okay, so first, I've mentioned California Riverside, Texas Southwestern are involved in this. We also work with Lauren Etheridge at Oklahoma University, Mike Ragazzino at Illinois Chicago, and we have collaborators assisting with EEG analysis at University of California, San Diego. I think the real key to our center is how do we integrate lab and human studies very closely with an eye toward improved treatment development, but also really focused on understanding brain mechanisms of pathology. So what is dysregulated? What is wrong with brain function in Fragile X in humans? And how is that best modeled and related to what can be studied in the lab in synchrony? So when you look at our projects here in our diagram, starting at the top right, we have human studies. So Ernest Pedapati is going to talk about our high density EEG, and here's a picture of an individual with Fragile X. Then they graciously agreed to use their photo showing getting EEG signals or brain electrical activity signals from individuals with Fragile X. And then in this context of studying brain electrical activity, we have the RX or the mortar and pestle here signifying that we are going to evaluate drug treatment, single dose drug treatment on brain electrical signals. And then also we expose individuals to various sensory paradigms, so sound and touch among them, and look at what happens to brain signaling with those exposures. And then in the middle, we really have the alive mouse studies. And what you see here is a mouse with a bunch of wires leading to the skull, and that's multi-electrode array technology, or that's the animal equivalent of high-density EEG studies. And then in a live animal with the Fragile X FMR1 gene knocked out, we can study EEG signaling, we can study the impact on signaling of drug treatment, and we can study the animal's response to stimuli such as sounds in an awake and freely moving animal. So that's the middle, really the bridge from the human to the mouse is tied together with oscillatory or brain electrical activity study and parallel study of drugs across that. And Devin Binder and Khalil Razak at University of California, Riverside are leading that effort. And then we move over to the left in our ex vivo neurophysiology, or essentially work in slice or probing at a microscopic and often cellular specific level, what is happening with neuron and neuronal network activity. And how does this relate then to the final box at the bottom left, looking at individual subtypes, such as parvalbumin and CCK neuronal species, and then also at that level of interrogation, being able to really drill down on mechanisms and neuronal signaling across cannabinoid signaling, GABAergic and glutamatergic signaling. So this is our overall design. It builds upon the success of the prior U54 NIH Center directed by Kim Huber and the successes we had there in synchronizing studies. But I think, the biggest take-home message is we're doing human, live mouse, and brain slice, sort of microscopic neuron level work that's tightly integrated to improve treatment development and brain understanding overall. So our first aim of the overall center is to identify biomarker measures and new paradigms suitable for a cross-species study, build upon what we have learned in the past and bring that forward. And by doing this, we're conducting neurophysiological or EEG and behavioral studies in humans with Fragile X and in mice, knockout mice that are alive and freely moving. And this is including some developments that Devin Binder will mention, but bringing the surface multi-electrode array also with a depth array to be able to do recordings deeper in the brain in the in vivo mouse models. And then at a neural circuit level, be able to interrogate and look at local and long distance circuit physiology in slice preparations. So here is a summary of kind of, what we've done in the past and what we're building upon. And the top panels from left to right represent looking at resting state gamma or high frequency oscillatory activity across slice preparation at the top left to in vivo mouse multi-electrode array to human studies. And really across these modalities or these means of investigation, we see increased high frequency activity. Again, really trying to demonstrate with real purpose here, synchronization of translational studies. So then if we use these markers in humans, we have a animal model that's better evaluable and we can extrapolate results back and forth. So overall aim 2, it's into testing on neurophysiology and cognition, targeting excitatory and inhibitory neurotransmission across glutamatergic and GABAergic pharmacologic probes. So this allows us to interrogate the impact on this brain oscillatory brain electrical activity using drug probes that modify glutamate and GABA activity in the brain. And this builds upon work we've done earlier, represented here in the prior center iteration with baclofen. And on the left panel, we see knockout mouse baclofen data where we see reductions post-baclofen in high-frequency gamma band power. And then in the right panel, we see a very similar finding in humans. And this is with single dosing, and then in animals went on to chronic dosing, but just with single dosing of 30 milligrams of baclofen. In 18 adolescents and adults with Fragile X, we saw gamma band rescue or significant downregulation of otherwise overactive high-frequency brain activity. Similar in the mouse and the human, synchronized data analytic pipeline, really trying to tie together the translational medicine story. So aim 3, test specific modulators of the endocannabinoid system on EEG biomarkers, behavior, and physiology in the Fragile X knockout mouse. And in doing so, really advance mechanistic understanding of the role of CCK positive interneurons and determine potential therapeutic effects of enhancement versus suppression of this signaling in Fragile X and identify the most promising candidates. And this work is extremely important also in light of recent results that came out from Zynerba Pharmaceuticals about transdermal cannabidiol that failed to show significance on primary endpoints, may have had a pre-planned ad hoc analytic finding related to a specific subgroup of patients. But again, as a field, we really need to sort out, what individuals with Fragile X may have cannabinoid signaling abnormalities and what direction on a more individual or patient subgroup level would we want to augment or suppress cannabinoid signaling. So we're really trying to get to the bottom of this, and I think that'll help interpret these results where we have limited understanding of a field of really interpreting what happened or how to best modulate cannabinoid signaling in Fragile X. So overall, and the way I think about this is, how do we change treatment development in Fragile X? Well, I think we do it by being mechanistic in our approaches. And when we study a new drug, we want to know what that drug may be doing to brain activity. And that can be in lab mouse studies and in human studies. And we want to use similar assays across mouse and human study to evaluate response. And at the same time, we need to improve treatment discovery in Fragile X syndrome. And I think our work in the cannabinoid system and looking at neuronal network signaling at long and short range levels will allow us to do that. And I think until we can best sort of mirror our early human experiments with treatment with what we've learned in the lab in synchrony, and we have tight rationale to what we are targeting with treatment, and we evaluate treatment effects similar in the human as we do in mice in the earliest human treatment studies, we may be destined to continue to have challenges in the Fragile X field. I think the final piece that this center, this group of investigators that I'm very lucky to be a part of and support the leadership of the group is who with Fragile X syndrome is best modeled by the knockout mouse at a physiologic brain activity level. And therefore, how can we best interpret early preclinical lab findings and extrapolate them into human study? I think we are really on to that. And our mechanistic approach in synchronizing lab and human science, I think, is essential to de-risking and improving our treatment development efforts. So with that, I'm going to stop my share and we're going to move on to our human EEG study work with Ernest Pedapati.
Yes, and I'd just like to introduce Dr. Ernest Pedapati, who is also a FRAXA grantee. We have a grant underway to study human gene therapy, but today Ernest is going to be talking about his work in electrophysiology, biomarkers, and pharmacology. So Ernest, take it away.
Oh, that's great, Mike. I appreciate it. Now, I turned off all my notifications, I thought, but then I got a notification for that there's free Big Macs at McDonald's today, so you can't escape into it. Let me start here. So my name is Ernest Pedapati. Hold on a second. Just get my Zoom bar here. I'm Ernest Pedapati. I'm an associate professor in the Division of Child Psychiatry and Child Neurology at Cincinnati Children's. I work as a psychiatrist for children and adults with Fragile X syndrome, as well as being involved in Fragile X research. Today, I'm going to discuss what I think are exciting results from the human portion of our program. The big picture of our research program is how to effectively measure brain activity to help develop new treatments. It takes a large interdisciplinary team consisting of physicians, psychologists, neuroscientists, and basic scientists. Craig Erickson and John Sweeney co-direct the Human Research Program. We have a number of co-investigators that are experts in specific areas. So my primary role is the supervision of the EEG participation and advanced EEG analysis. Lauren Schmidt is a psychologist who specializes in neurocognitive studies, including combining EEG and behavioral tasks. Lauren Etheridge is also a psychologist, and she is an expert in studying EEG related to sensory systems, including auditory and touch. Dr. Rebecca Schaefer is our lead psychologist who coordinates all of our psychological testing. And Christina Gross herself is a well-known Fragile X researcher, but has been gracious enough to join our program and provide specific expertise in blood and molecular biomarkers. Stepping back, let me give you a big picture view of what we're trying to accomplish. As physicians and psychologists, our bread and butter is studying behaviors. We make decisions based on what we see and hear from patients and families. But of course, these behaviors come from the brain. To understand this black box, we need to measure brain activity in detail. But why not just rely on behavior? It's because the treatments we are using work on the brain. So unless we can match the treatments with brain activity, we can't know for certain what we're testing and treating. Now, this is not that unusual in medical research. For example, breast cancer research made the biggest leaps once they could stage, size, and genetically identify tumors. In Fragile X, of course, we have had many drug trials, but very few successes. We think this is because we are able to do a good job of classifying behaviors, but not a very good job of classifying the underlying brain activity. But not just any biomarker will do. In Fragile X, we have a genetic test. So any new biomarker that just shows a difference between someone with and without Fragile X is not going to move the field forward. This biomarker also has to reflect the core features of Fragile X, not just co-occurring symptoms. And it must be useful to both predict and track changes to treatment. And ultimately, it must be clinical grade. And by this, I mean it is good enough for physicians to use in daily practice, such as ordering thyroid tests. Over the last 5 years, we have worked hard to collect as much brain data from participants with and without Fragile X to begin the process of developing just such an advanced biomarker. Just like in an earlier slide, we work to make the best description of a patient's behavior and then get EEG activity both when the participant is resting and watching a silent video, but also when they are listening to specific patterns of sounds. Ultimately, our data set includes 73 individuals with Fragile X syndrome and 71 controls, which is the largest, if not the largest collection of high-dense array EEGs in a rare neurodevelopmental disorder in the world. This is a brief talk, so I want to focus specifically on one particular type of brain activity because we can measure it both in mice and humans. Gamma activity are waves of brain activity that fluctuate over 30 times a second and seem to come in two different forms. One form is during general brain activity. We call this broadband gamma activity. But there's another type of gamma activity that is also very fast, but very narrow in a specific frequency band. The video you're watching right now is what broadband gamma activity looks like, measured from a participant in our laboratory. This is a great example of broadband gamma activity. This is what narrowband gamma activity looks like. If you look at box number 1, that is the sound pattern. Number 2 shows 2 blue boxes. The typically developing subjects are on the left, and the average of Fragile X subjects are on the right. The bright red is the specific task-based gamma activity, that narrow band right in the frequency range of that stimulus. If you look at box number 3, this shows that Fragile X subjects have much less narrow band gamma activity. That's represented by the blue. And in box 4, they have much higher broadband gamma activity. We think of this narrow band gamma activity as functional gamma, or gamma that is useful for specific behaviors. When we first started, we had a simple hypothesis. Looking at number 1, we see that our earliest EEG findings measured from the scalp, we expected it to predict intellectual disability. Our idea was that broadband gamma activity, the more you had, the more to interfere with cognition. Our initial efforts were disappointing. When we looked at the relationship between scalp EEG activity and cognition, there was no relationship. The plot in figure 2 demonstrates that as gamma power increases, you cannot really predict intelligence score. So stepping back, this was not very useful. We have to think about new ways of analyzing data. When you take EEG data straight from the scalp, it's contaminated by scalp muscles, and this is not a very good representation of brain activity. So we performed a technique called source analysis, where we transformed the data based on an MRI image to predict what parts of the brain this EEG activity was coming from. And you can see the procedure here. Following this process, we now had a very different form of data that in theory eliminated much of the contamination. If you look at box number 1, this is a visual representation of where we found the increased broadband gamma activity affecting the temporal cortices of the Fragile X brain. Now, if our theory was correct that broadband gamma activity was related to cognition, we are now more confident that the signals actually represent brain activity. We would now expect to find a much tighter relationship. Even though we expected it, we were still surprised at how much of a difference the source localization techniques made. Now the relationship between increasing gamma activity and decreased cognition was much more clear. Box 1 shows this relationship between all subjects, including females and mosaics. However, on the right box, you can see that the full mutation patients seem to drive this relationship the most. This relationship of 0.6 is very significant and represents the strongest correlation between intelligence scores and EEG biomarker to date. We took an alternative approach. Now that we could model activity within the brain, we began to group certain regions to form functional networks. In particular, the default mode network is associated with brain activity, that when you are not doing a specific task. We reasoned that how the brain was doing at rest would likely be predictive of cognition as well. The other advantage this has is that when you model a network, you no longer need a comparison group. So we could get values directly from a Fragile X subject without needing to have a contrast with a control. What was remarkable was that we found massive changes in the different functional networks in terms of gamma activity. Not only was the default mode affected, but the functional networks including emotion, attention, and sensory networks including vision and sound also had increased gamma activity. Now we could make a similar conclusion of the relationship between broadband gamma activity within different networks and cognition. Again, this relationship was primarily in full mutation males. And so here you are seeing the relationship between gamma activity. The more you have, the more severely affected your intelligence is. But as you know, some females with full mutation Fragile X are more severely affected and similar to males. So what would happen if we examine the relationship between gamma activity and cognition, not based on sex, but rather based on clinical groupings? Our data set was large enough to blind separate groups based on clinical symptoms using machine learning. We found 2 groups, which is not surprising. One group that was more severely affected, cluster 2, with more psychiatric symptoms and decreased cognition. But in this case, the groups consisted of males and females. This was reassuring, as we found that severely affected individuals, regardless of sex, showed a particularly strong relationship between cognition and broadband gamma, as you can see in box number 1. This relationship was not present in the mildly affected individuals. This raises an important question, because when considering that full mutation males have a similar relationship, it may be more about the distribution of protein in the brain that affects gamma activity than simply your biological sex. This is a talk about gamma, but I want to show you one piece of data in our most recent analysis that is raising some very interesting questions. There are other brain activity bands, such as alpha activity, which is associated with inhibition with other types of brain activity, including gamma. Here in the red box, you are seeing an incredibly tight relationship between the amount of alpha inhibitory activity and how much anxiety the patient reports. This is particularly exciting for us because we have a potential new brain areas and disease-relevant biomarkers that fit the tough criteria we were discussing at the beginning in terms of biomarker development, and that's in the red box here. So to summarize this model, when there's FMRP loss, 2 things change. We find both decreased inhibition and increased excitability. This shapes the types of brain activity that the brain can produce. In our model, this shifts to more broadband gamma activity in Fragile X, but less narrow task-based gamma activity. This would lead to changes in functional networks and also display symptoms. Now, in humans, we can draw attention to these associations, but we cannot truly say if the cognitive changes cause the increased gamma or if the increased gamma causes impaired cognition for sure. That is where our partnership with basic science laboratories who can answer this question can really shine. I want to thank you for your attention and interest in this work. Certainly none of this work is done alone. We are extremely appreciative to our research team and the participants and their families who have been involved in this project. Thanks very much.
Thanks very much, Ernie. That's really, really exciting. And as you can all see, the plot is thickening all the time. And so I'd like to move to our next speaker, Dr. Devin Binder, who is another FRAXA grantee at the University of California, Riverside. He's going to be talking about network mechanisms of neurophysiology and behavior in mouse models of Fragile X syndrome. You may recall that Devin actually presented some of the work he's doing on essentially animal EEG several sessions ago during our FRAXA seminar series. But I think this is important work and it deserves as much attention as we can give it. So Devin, I'll turn it over to you.
Sorry, unmuting myself would help. Sorry about that. It's a classic Zoom move there. Can you hear me okay now? Thank you. Well, I assume you can because I saw a facial reaction. Thank you. Well, anyway, I really appreciate the opportunity to be here again. It's really a privilege to be involved with this group. For several years, we've been working together also with my colleague, Dr. Khalil Razak, on basically collaborations on both sides with the Cincinnati group and also the Texas group. You'll hear from Kim Huber in a moment. And our role, as of course is mentioned by Craig, is to look in the mouse model of Fragile X and see what we can determine in terms of both EEG and behavior. And so our topic is termed network mechanisms of neurophysiology behavior in the mouse models. And I'll run through this relatively quickly because it was already introduced by Craig and Ernie. We were, of course, stimulated a few years ago by the presence of these remarkable EEG power differences in the Fragile X patient population. And also this interesting issue of response to auditory stimuli. So this is the so-called chirp stimulus, where there's a reduced phase locking. In other words, a 2-second modulated broadband noise that you can see here. This is what's called an up chirp. And the normal control response is that the brain synchronizes with a certain power and frequency dependence over that 2-second or 2000 millisecond chirp. And the Fragile X patient population has in a lesser response, a lesser synchronization or reduced synchronization of the response. So this is a different parameter that we can use in addition to the differences in baseline EEG power. So we started with this based on the human data, which was just shown. Lauren Etheridge and others created those data, this is from Molecular Autism in 2017. Stimulating by those data to try to develop measures in the mouse that would also correlate, as Craig said, to try to develop what you might call translational EEG biomarkers. And most recently, and as I'll describe, we've been interested in trying to do this with multi-electrode arrays to more closely approximate the human condition where the human scalp is covered by 128 electrodes in some montages where they can, as Ernie has shown, can get really interesting derived EEG data in multiple, not only across frequencies, but across brain areas. And we wanted to move from 2 electrode studies in the mice to the multi-electrode array technology. And then, of course, the translational aspect of this is to try to use these EEG sort of readouts in the mouse model to look at the effects of specific pharmacological target manipulations. As I'll describe, we have some targets that we're looking at in this particular group project. So Johnny Lovelace was a postdoc in our laboratories, combined between myself and Dr. Razak, Khalil Razak. Several years ago, he started these studies, and this is a background to where we are today, that with two electrodes, one in the auditory cortex and one in the frontal cortex, in the Fragile X knockout mice, we could indeed see these phenotypes of increased gamma power at rest. And then on the bottom, you see the chirp response being partially abrogated or reduced in the knockout mouse. The right is just a knockout minus wild type with some statistical analysis of areas that are different. In other words, we were able to see the gamma phenotype and the chirp phenotype in the mouse in the two electrode situation. Kerry Jonak joined my lab a few years ago and what has been instrumental in developing multi-electrode arrays to try to extend and really deepen these findings. And the idea in these experiments is that we're actually implanting a surface, planar surface multi-electrode array, which is microprinted with MEMS technology. And this is made by, in our case, NeuroNexus. There's many companies that make these probes these days. And Kerry implants these in a very specific orientation so that bregma in the mouse, which is, of course, the confluence of the coronal and sagittal sutures, so that we can implant in multiple groups and compare the local spatial localization of these with a recognition of the caveat that there could be volume conduction through the skull. So we're always thinking about those issues. But what was immediately noticeable when she did this, even a few years ago, was that there's this remarkable difference in resting EEG between wild type and knockout mice. As indicated here, the more sawtooth pattern basically indicates a high gamma power. And in fact, if you then analyze this, and these are the mice, they're freely moving after, and so we're trying to get as much naturalistic an environment as we can. Of course, this isn't exactly naturalistic, but the mouse is freely moving, and then this is in a sound chamber where we can play the auditory stimuli such as broadband noise and chirp, and look at event-related potentials and various stimuli. So we see that indeed there's this remarkable difference in resting EEG power, and in fact, it's across multiple frequency bands, not only in gamma but in lower frequencies. And then we're also able to get with this regional differences. So you can group electrodes in, for example, left or right frontal, left or right medial, which is sort of parietal area, and left or right temporal areas, and you can see region-specific changes in the EEG. So at rest, we saw this. This is relatively recently published data, but now reproduced multiple times. This shows that there's this increase in power in the knockouts. Well, the other thing that Kerry did was found that there's indeed a remarkable change in the chirp response. And this is really across the brain, although it's interestingly different in magnitude in different areas. But basically, throughout the brain, there's this remarkable difference in the chirp response. So we wanted to use these findings to demonstrate that we can implement the EEG, the multi-electrode array EEG, and reproduce these phenotypes that were seen originally in the two electrodes. And most importantly, of course, that those phenotypes are similar to those that are observed in the patient population. Again, along the lines of what Craig said, we want to tightly integrate what we're doing with our Cincinnati colleagues. So in conjunction with that, we are now actually directly taking the multi-electrode data and processing them with Cincinnati as part of this group project in similar ways so that Ernie and his colleagues can process mouse EEG alongside human EEG for various types of derived parameters. And I mentioned actually in the August presentation that I did here for FRAXA that we've had now some data, which Craig actually mentioned, and he mentioned it alongside the human data, that single-dose acute racemic baclofen administration actually dose-dependently suppresses EEG power, and yet it also increases phase locking to chirp stimuli. So in terms of a potentially salutary or improvement in these so-called EEG biomarkers, we see that with the single-dose racemic baclofen, as Craig said. So we're doing that alongside the human data, again, as part of this tight integration. So in this particular project going forward, as part of this NIH Center, we have several specific aims that I want to just briefly mention. So again, in coordination with Project One, which is our human colleagues that Ernie and Craig mentioned, we're going to take these MEA data, multi-electrode data from both adult knockout versus wild type mice to derive further translational EEG biomarkers. So we want to go beyond gamma and chirp. We want to look at connectivity phenotypes and other parameters, as I'll describe in a second. We want to also extend these findings into developing mice. This is a really important issue that, when do these cortical EEG abnormalities, and we don't know they're cortical, when do the EEG abnormalities develop? So we're going to apply the MEA to P23, 24 mice and develop that. And then really interestingly, we're going to apply depth multi-electrode arrays, which are now available. Stereotactic implantation of these depth MEAs can be done in a similar way. We can now get depth specific or layer specific cortical physiology, and that's in progress. So we want to do that as our next part of this project. And then, of course, the structural aspect of this is that we want to conduct these EEG analyses in parallel with the similar algorithms that are used in humans, so that we are using a validated analysis pipeline. And that includes important issues such as artifact rejection and principal component analyses and things like that. The idea being that we would identify not only these power-related and chirp-related, stimulus-related dysfunctions in the Fragile X mouse EEG, but also compare it to the human data. And then with the depth to identify any type of laminar specific changes. So these are some of the parameters, for example, and they're not limited to these, but these are some of the parameters that we are considering and in the process actually of with Ernie developing with the mouse EEG, and that is coherence among brain areas, something called debiased weighted phase lag index, which is a measure of phase synchronization among brain areas, and spread of coherent activity across brain areas, which there are data from John Sweeney and Craig Erickson earlier that is abnormal in Fragile X humans, in Fragile X patients. Principal component analysis, band-to-band coupling, such as theta gamma, alpha gamma coupling, and phase amplitude coupling are all derived EEG parameters that we can get from the MEA that we could not get from 2 electrodes in the mouse. So we're trying to validate and develop these in the mouse. Now, not once we've done that, but in parallel with doing that, we want to test, pharmacological target drugs or pharmacological targets with specific drugs as follows. So we want to test GABAergic, glutamatergic, and endocannabinoid mechanisms. In this particular aim that's on the screen, we've identified, and we're flexible on this. In the mouse, we can do this, flexibly depending on what our human colleagues can do. But for example, based on our racemic baclofen data, we are very interested in the R-baclofen, and I note that Mark Baer and his colleagues have recently stimulated more work in this area, which is of great interest to us, and we want to share all these data with that group as well, in terms of trying to see what we can get out of the R-baclofen. But Craig already showed the racemic baclofen data. We want to do that, and we want to do other drugs. This BAER101 is a GABA A agonist. The R-baclofen is the GABA B agonist. So we want to look at various target mechanisms here in the GABAergic system, and then also in glutamatergic system. We have great interest in some of these Genentech drugs. So GNE0723 is a drug that's a positive allosteric modulator for a glutamate NMDA2A receptor, which is thought to actually partially activate PV interneurons, which are known to be hypoactive in Fragile X. So there's some interesting mechanisms here that we want to probe. Memantine is an NMDA non-competitive antagonist that has some data in Fragile X. So we're going to try to test all these. And the idea is that we can do both acute single dose treatment, but also chronic treatment to look at issues of tolerance in the mouse in terms of the effects on EEG. And just briefly, I think I mentioned this in August at our seminar, but we see a very different effect of acute versus chronic baclofen in the racemic baclofen data that we have thus far. So we want to be able to assess both acute and chronic treatment with the drugs in the mouse EEG. In parallel, my colleague, Dr. Khalil Razak, is doing behavioral testing, and we're working together with Mike Ragazzino, as Craig mentioned, who's an expert in this reversal learning paradigms, which he's found to be very different between wild type and knockout mice. So of course, we want to see not only the effect of these drugs on EEG, but any rescue of behavior, particularly this reversal learning behavior, which appears to be behaviorally relevant in Fragile X. So that's the idea there. And then the endocannabinoid part, which is our so-called AIM3, is really interesting because, as Craig mentioned, there's a lot of there's a lot of studies, as many of you know, that Fragile X families are using CB1 or cannabinoids and cannabidiol study that Craig mentioned. So we want to look at target engagement on the endocannabinoid system as well, and particularly focusing here on CB1 receptor inhibition with some of these drugs. And there's some data already from some of my colleagues in this group, and also other data in the literature on some of these drugs that are CB1 receptor, either negative allosteric modulators or antagonists. So bottom line is we want to also look at and one thing I should mention is Kim Huber, who's about to talk, her group has generated these CB1 receptor knockouts. So we can look in a mechanistic way, in particular neuron cell types, as to what's the effect of knocking out the CB1 receptor on the cortical physiology, and the MEA EEG, combining not only with the surface, but also the depth physiology. So that is sort of a sampling of what we'd like to do. We want to work on, expanding our translational profile of EEG biomarkers, and that's sounds. I think it's only possible because of the nature of this group working directly with the human EEG and the mouse EEG together. And then, of course, identifying these MEA phenotypes in the developing mice, which we haven't done yet, and analyzing layer-specific physiology. And then, of course, in parallel with that, trying to look at drug targets. So this actual work is being done by my colleagues at UC Riverside. Johnny has moved on. Patty is our current postdoc. Carrie is our research associate and lab manager. And Khalil and his laboratory also have a number of people working on this project. This is just a picture from 2017 in the previous iteration of our group. But we're so pleased to be able to be involved with the support of both NIH, FRAXA, and with DoD. So thank you for your attention.
Thank you very much, Devin. That's really exciting. And I certainly don't ever get tired of hearing more about this. I think that's really the way to go in Fragile X research.
Thank you, Mike.
Our next speaker probably needs no introduction. Dr. Kim Huber is a superstar in the field. And I don't mean to embarrass you, Kim, but we've been working with Kim for a really long time now. She was a postdoc in Mark Baer's lab back in 1999 when she got a grant to study metabotropic glutamate receptors in Fragile X, and of course the rest is history. Kim is now a professor of neuroscience at UT Southwestern in Dallas, and she's continuing to work on Fragile X, and she is still a FRAXA grantee and studying all sorts of aspects of the electrophysiologic function of Fragile X neurons. So today she's going to be talking about FMRP regulation of local and long-range neocortical circuits in the mouse and how that links with EEG phenotypes. Kim?
Thanks, Mike. I guess I'm trying to figure out how to share my screen here. Oh, there it is. Okay, yeah, okay. I have done this before, once or twice. Okay, there we go. Okay, so I guess you guys can see that. Let's see, just want to fix my pointer here so you can see my pointer. Okay, can you see my screen and my pointer there?
Yes, we can.
Okay. So yeah, thanks, Mike and Katie. And yes, I basically got my start in Fragile X because of FRAXA, working as a postdoc in Mark Baer's lab. And so, yeah, I just really want to say that I think my own entry into this field, as well as I think many other Fragile X researchers, can be attributed to FRAXA. So you guys have made an incredible impact in the field and in your support of research and discovery in this field. So, and thank you so much. So I want to talk about what is affectionately known as Project 3 in the Center Grant, and I'm very excited to continue our work with this group. And this is a project that's co-led with my collaborator, Jay Gibson at UT Southwestern. And our focus really is to try to, a big question that you've already, heard a lot about from Ernie and Devin is these really remarkable, kind of EEG phenotypes that have been discovered in human that, as Ernie showed, have very good correlation with some of the behavior, symptoms, IQ in humans, and that, through a lot of hard work in the last center, Ernie and Devin and Khalil and John Sweeney have all discovered these common EEG phenotypes in the human and mouse. And I think that this gives us, a very important, question that I think, kind of arose with some of the past failures in clinical trials is, how much does the mouse model of Fragile X, really model the human disease, and are the mechanisms of dysfunction in the mouse conserved in the human? And again, what we understand and treat in the mouse, does this have links with the human? And so this piece right here is showing that we can at least identify some common EEG phenotypes in mice and human with Fragile X is very key. And so we want to utilize these kind of common, EEG phenotypes seen in the human and mouse to try to understand brain dysfunction in Fragile X and, and identify potential therapeutic targets to improve or correct brain function. So Project 3 links in here where we're really trying to understand the cellular, synaptic, and circuit mechanisms that contribute to this EEG phenotype, which in turn, we think then links with humans. And so the goals of Project 3 are to understand synaptic and cellular circuit mechanisms that correlate with that underlie the mouse EEG phenotypes that are relevant to humans with Fragile X. And by this understanding, we hope to identify as well as test therapeutic strategies to improve or correct circuit dysfunction, and in turn the EEG, which may provide evidence that these therapeutics may be effective in humans. And then in our work, I think we hope to identify the cellular and developmental loci of FMRP function to both understand the cell-specific roles of FMRP and the direct and indirect roles, which I think is important in understanding, kind of how loss of FMRP leads to the dysfunction of the brain, as well as, again, to identify therapeutic targets. So if you've already heard, just to remind you, is that one of the kind of common EEG phenotypes that's observed in both the human and mouse EEG is this enhanced resting state gamma power, or as Ernie was referring to as broadband gamma power, that's observed in both mice and humans. And also, as Ernie suggested, that this is indicative of hyperactive or hyperexcitable local cortical circuits and deficient, inhibitory circuit function. Another phenotype that's seen in the humans in the EEG, as Ernie referred to, is this impaired network function, which really refers to, potential abnormal or deficient long-range cortical interactions, potential cortical connectivity and function. And as Devin talked about, one of their goals is to try to do these types of analysis in the mouse EEG to see if there's similar network dysfunction in the mouse. So we have data that we put in this grant application and that we continue to work on of, I'd say, cellular and synaptic mechanisms that may underlie hyperexcitable local cortical circuits, as well as abnormal long-range cortical connectivity. And we hope to, by leveraging this kind of mechanistic knowledge that we have, to try to link these more local circuit or synaptic changes that we see to EEG phenotypes through collaborations in this center. And so just to give you a kind of a schematized version of what we see, we've been doing a lot of work in the somatosensory or barrel cortex, which we're kind of using as our canonical cortical circuit. And one of the kind of key, kind of robust phenotypes that we've seen is evidence of this hyperexcitability. And if you do recordings, electrical recordings of cell activity in these cortical layers, and these are all in acute brain slices. These are about 3-week-old, adolescent mice. What we see is that in wild type normal mice, you can see these bursting patterns of activity, but in the Fragile X syndrome mice, when this activity occurs, it's prolonged. And basically these, our conclusion from this is that when these circuits are active, they fail to turn off or turn off at the right time. And there are correlates in vivo of sensory evoked from Khalil Razak and others, that there's also this kind of hyperexcitability or prolonged circuit activity that's seen in the Fragile X mouse. And we've also identified some cellular correlates of that would be expected to cause hyperexcitability that may be contributing to these long activity states. One of these is hyperconnectivity of these excitatory pyramidal neurons, specifically in layer 5. So we see in FMR1 knockout mice, as we published several years ago, that these neurons are hyperconnected. They have more local connections, and we've shown that this is due to FMRP functioning in these postsynaptic layer 5 pyramidal neurons. So this is a local cell autonomous effect of FMRP. We've also identified dysfunction of 2 inhibitory neuron circuits, basically deficits in inhibitory neuron circuits. One is there's reduced excitatory drive of these parvalbumin positive inhibitory neurons. And then secondly, we and others actually bred aldosterone in the hippocampus, and we have data in the cortex to suggest that these cholecystokinin neurons, which are known to be very sensitive to endocannabinoids, that these have an enhanced endocannabinoid mediated suppression of their function. And so we have these kind of 2 different inhibitory neuron circuits that have reduced function, and our hypothesis is that these are contributing to the increased resting state or broadband gamma in Fragile X. And they may also be contributing to this abnormal sensory evoked or functional gamma. So in our grant proposal, one of the kind of questions that we want to ask is we're going to focus on these 2 inhibitory neuron circuits and ask, does dysfunction of specific inhibitory neuron circuits contribute to this local circuit hyperexcitability, the abnormal EEG, and these cognitive flexibility or probability learning in the Fragile X mouse model? And so questions, that we can address is, do specific inhibitory neuron types, either CCK or parvalbumin, do they contribute to different aspects of the EEG phenotype? And is targeting or correction of one inhibitory neuron circuit more therapeutic than the other? And this will be done in a collaboration we're going to be working in acute brain slice and microcircuits, and we'll be collaborating with Devin and Khalil on in vivo mouse EEG and mouse behavior. And specifically, as Devin has already introduced, because of our findings with these endocannabinoids and the CCK neurons, we're going to be manipulating endocannabinoid receptors and specific inhibitory neuron types, and also using pharmacological strategies to affect the endocannabinoid system. And we're hoping that this can address some of the kind of confusion in the field where there's evidence that either enhancing or reducing endocannabinoids can be therapeutic in Fragile X, and we think this may depend on the cell type that's being targeted, and we hope to provide some answers to these questions. And then we are also going to be targeting these inhibitory neuron types, the cholecystokinin and parvalbumin, using optogenetic, chemogenetic, and novel pharmacological strategies to manipulate and correct their dysfunction to determine how these are contributing to the circuit excitability that we see in brain slices, as well as the EEG phenotypes observed in vivo, again, in collaboration with Project 2. Okay, and just in the last few minutes, I wanted to talk a little bit about some of our new data where discussing really the effects of FMRP and Fragile X syndrome on long-range cortical connectivity. So we know that cortical neurons can connect to their neighbors in a local circuit, really within a cortical region. But we also know that neurons can make monosynaptic connections across brain regions, such as between sensory and motor areas, or sensory and higher order cortical areas like frontal cortex, and can also connect between hemispheres. So probably most of you know that the corpus callosum is this major, axonal white matter tract that connects the two hemispheres of the brain. And importantly, from structural imaging and functional imaging in humans, it's known, and there's suggestions out there that there's imbalances between local and long-range connectivity in humans with autism spectrum disorder, as well as Fragile X. And there's recent data in the Fragile X mouse that there's actually reduced corpus callosum volume with using diffusion tensor imaging, and also reduced kind of functional connectivity between hemispheres using functional MRI. And there's recent data from, I thought was a very nice study in JAMA Psychiatry, showing that there's reduced corpus callosum structural integrity in infants with Fragile X, observed as early as 6 months of age and persists up to 24 months of age, which is latest they did in the study. So suggesting that there's abnormalities in the development of these corpus callosum long-range connections that are, connecting the two hemispheres. So we've set out to try to understand, is there reduced structural and functional connectivity? Is this mediated by synaptic connectivity of circuits, long-range circuits in the Fragile X syndrome mouse model? And then to understand the cellular, molecular, and synaptic mechanisms of this. And one of the ways that we've looked at this is we can label neurons by injecting an adenovirus expressing channel rhodopsin, which is a light-activated ion channel, and a fluorophore M-cherry. We do this in a postnatal day 1 mouse. And we can label these neurons that then project their axons to the other side of the brain. And then at about 3 weeks of age, we can cut a brain slice from this hemisphere and image it, and we can see these fluorescent axons that are originating from the other side of the brain. And then we can probe these synaptic function from these callosal axons by recording from here. It's a layer 2-3 neuron in the somatosensory cortex, and we can flash the brain slice with blue light to elicit neurotransmitter release from these axons. And we can do this either looking at the whole, activating around the whole cell, or in specific parts of the cell to look at the subcellular localization of any callosal axon activity. And so just the bottom line here is that we see weak callosal inputs onto layer 2-3 neurons. That also, we see this in layer 5 as well. And this is consistent with the reduced corpus callosum volume that's observed in the Fragile X mice. And we also observed that this is really localized to the primary apical dendrites of these layer 2-3 cells. So kind of putting this together, we have these kind of hyper-excitable local circuits that I've talked about earlier, but when we look at long-range connectivity with other circuits across the brain, and specifically we've looked at the contralateral somatosensory cortex, these long-range projections are weak. And they may also be weak with other cortical areas, and this is one of the goals of the grant. We've done a lot more work on this. We know that these synapses in the FMR1 knockout They're actually there, but they're immature, what we call silent synapses, and that they don't have AMPA receptors, but they have NMDA receptors. We know that it's actually mediated by postsynaptic FMRP in these pyramidal neurons, in layer 2, 3, or layer 5 pyramidal neurons. We don't think it's actually due to FMRP in the callosal projecting presynaptic cells. And importantly, these are sensory experience dependent, that FMRP is interacting with experience to regulate development of these long-range connections. So we think that this may underlie, some of these impaired functional networks, and that Ernie has identified in humans, and may lead to abnormal sensory processing or sensory integration. Sorry, something's popping up here. Okay. So just the aims related to this work is we want to understand the mechanisms and functional consequences of these weak inter-area connections. This is really new data. We know very little about it. We want, to look at, we want to understand how these long-range connections regulate local circuit activity. We want to examine connectivity with other cortical regions, and then we are also going to look at the ability of postnatal reexpression of FMR1 to restore these long-range connections. And because we know so little about the mechanisms of this, and to help to us identify the understand mechanism as well as therapeutic targets, we want to, and we know this is due to FMRP in these layer 2-3 glutamatergic neurons. So we are going to do some molecular analysis of both the transcriptome and the translatome in these cells with and without FMRP, and then also to understand how experience regulates these molecular changes. Okay, and just say we have a lot of work to do. We really need help. So we are hiring, and if anyone's interested in postdoctoral positions, please contact Jay or myself, and we're also going to be posting this on SFN NeuroJobs hopefully soon. Okay, and I'd like to acknowledge the people that have done this work. Jay, as well as Zhejiang, did all of the long-range connections, and then Daria Gonzalez and Gemma contributed to the local circuit hyperexcitability work. So thank you for your time. I'll take questions, or we will all take questions now, I guess.
Wonderful. That was a really great talk, and it's a really exciting project. You're not kidding. There's a lot of work to do. Big project. I think that's really great.
Yeah.
Well, so now we have plenty of time for questions. And as I mentioned before, the way to ask a question is to go to the chat function and ask your question. And if there's someone in particular you want to ask the question of, you might want to just note that. We have our chat table?
We have not received any messages yet.
Ah,
That's new.
yeah, usually we have lots of questions by now, but obviously people are just dazzled by the brilliance of these presentations.
Here's
For Kim.
Okay. Okay.
How early can you see the hyperexcitability in slices?
Yeah, that's a good question. We really have only looked as early as like P18, but we postnatal day 18 in mice, but we know that it can occur as late as like P60, and then we've recorded these excitability in vivo in older animals. But we are interested in going younger as well, because I think that for kind of technical reasons, we can study these individual cells and their contribution. So we are planning on looking earlier, like P14. Yep.
Junyu Zhao wants to ask everyone, but I think probably especially Craig and Ernie, which drugs have been shown to exhibit different responses in people with Fragile X with high versus no gamma power, or high versus normal gamma power, I would
So we continue to sort through this. We're currently evaluating gamma differences with treatment supported by FRAXA of a drug called AZD 7325, now called Bear 101, which is a GABA A subtype receptor alpha 2/3 agonist. And we are evaluating their correlations with gamma change and clinical improvement. In the last center iteration, where we had the opportunity with this group, we looked at single dose lovastatin, minocycline, acamprosate, and baclofen. And baclofen had the most clear impact on gamma oscillatory activity in humans. And as I said, also was noted in the mouse. We saw less, clear signaling with resting state EEG change with those other drugs, albeit only with a single dose. We're using this technique in idiopathic autism too, and we've been studying minocycline in autism, and we're analyzing data now to look at resting state change. In that project in idiopathic autism, we only enrolled folks with high resting state gamma activity, and that's really Fragile X findings, kind of carrying over into other areas. So we're evaluating a lot of this data now. I think I would hypothesize that our small molecule therapy in humans is looking to potentially be most successful in full mutation males with no discernible protein, with the highest resting state gamma abnormalities or the most dysregulated gamma at rest. I think that as we match up small molecules to those profiles, we will use that to parse the groups and evaluate target engagement.
Okay, a question for everyone. Great session. We heard a lot about the mouse, but can you comment on the status of the rat model and its EEG phenotype? How's the background strain of the mouse influencing this EEG phenotype, or is it stable between different strains?
That's a great question, if I can take that, and I look forward to my colleagues' comments as well. We thought a lot about Fragile X knockout rat model, that it exists and that it'd be worth looking at the EEG, and then someone did it, and it's just been published very recently. We could try to provide the link. I have it. Was one of my colleagues sent it, John Sweeney, and also. But the bottom line on the Fragile X knockout rat, it's not our study, but the one that was just published, really is quite remarkably confirmatory, and we're thrilled to see that the rat has the gamma power phenotype, and it also has the chirp phenotype, meaning reduced synchronization to repeated sounds. So the details would be in their paper. That's not our group, but I'm happy to try to provide the link for any observer to this broadcast. But that's a great question. And the second question about mouse strain is a very important question. We did some initial studies in C57 versus other strains, and we also have this issue of, when you get mice from jacks versus when you've bred them in your colony for a while. So all those types of things do influence actually baseline EEG power. But we've seen a difference despite the strain if there's a Fragile X knockout. So within strain comparison, there's the same differences. What we've been publishing on is C57s, just for whatever reason. That's what we've settled on, various reasons for that. But bottom line is, yes, the rat data have been published. It shows the similar phenotypes, so we're thrilled to see that. And number two, it's strain dependence definitely there for the EEGs.
Okay, Julie would like to know if there's any data in humans or mice about what happens to the EEG phenotype during development.
I'll quickly comment on the human. So, our data set does have a number of ages in it. There's some very traditional EEG findings that even change in typicals over time. And so we are working on trying to be able to answer that question, but we really need a good control group to be able to understand that. But certainly we do feel like there are some pretty important insights to be drawn from that.
If I could just extend that question a little bit, Ernie, a couple of sessions ago in the seminar series, we heard from Carol Wilkinson and Chuck Nelson, and they were talking about their studies in very young children with Fragile X, and especially correlations with language development. And they found a pretty good correlation between better functioning and higher gamma, which on the surface of it would seem to be at odds with what you folks are finding in your link between IQ and gamma function. Could you comment on that? Because I usually these apparent discrepancies are really not discrepancies at all, and they're really just informative. But could you comment?
Yeah, it's a good question, and I think hit some of the other questions that are in this chat here. That there's a complex interplay here. It's more than just increased gamma, but it's the relationship with the other bands. And what we realize is that just like gamma, you can have high task-based gamma, which is extremely adaptive, versus high broadband gamma, which is not so much. And so there's probably some balance between the interference of broadband gamma along with the ability to be able to have that signal to noise ratio, to have the types of connectivity networks to really focus on a particular aspect. Now, what's nice about young children is they learn, just from a clinical perspective, there's so much learning that goes on during those stages. And we would expect there to be lots of gamma activity just in terms of different brain functions working and coming online. And so it's an important question, but one that really has to be narrowed down both on the relationship between the lower bands and the higher bands, what we call phase amplitude coupling, which is the way that the brain sort of communicates these long distances, and looking at this discrepancy between narrow band and broadband gamma.
Mike, also, I don't think I featured it adequately, but in the human work in this center, we are doing developmental EEG work. And that a lot of what Ernie talked about in our strongest findings are in more of an adolescent through young adulthood age range. And a lot of our early treatment work with EEG outcomes has also been in that age range. But we're doing developmental youth EEG studies. And Devin mentioned he's doing developmental mouse studies because it very well may be that the best neurophysiologic markers may differ across age bands and development. So you could even see something like a gamma activity arc change, and that needs to be well understood because we would be looking to essentially, for lack of a better term, correct different things at different ages as the brain presents abnormalities of neuronal signaling. So I'm glad you mentioned that, and we're beginning to do that, but that's going to be essential, especially as we get into infant and toddler and younger child therapeutics, that the same approach to evaluating target engagement may not hold.
There's a really nice question here from Arjun in the comments. And, what Craig and I are talking about is that there's a real physiological link here. Like, for example, the visual cortex, we know there's certain pathways that are particularly rich in FMRP. So, for example, in our data, the strongest clinical correlations we have are with the visual network gamma and clinical findings. And so they're really, this is hopefully will be tied down to that real base level on where is FMRP being distributed, and is excessive broadband gamma activity a property of networks that are FMRP scarce, for example.
And if I can add to that, Ernie, that's a great question from Arjun. Just to repeat it for those who didn't see it, how do you think about the disparity between local and long-range connections? Discussions with Kim Huber have really kind of crystallized this question to me as well in this context. I would say the other thing, just to mention on the mouse side, we are starting just to analyze, and we're having research meetings even this week about this, of how to analyze pairwise comparisons in the mouse MEA from local versus long range. And specifically, as Kim knows, we talked about this last week, we're trying to develop ways in which we can analyze, intrahemispheric connectivity versus interhemispheric connectivity. For example, homologous sites in the temporal lobe across the corpus callosum in, let's say, A1, the equivalent of auditory cortex, although we can't precisely localize that with surface EEG. But for example, like, what is the connectivity between hemispheres versus intrahemispheric? And in fact, we want to see if we can differentiate those issues in the mouse as well, with the huge caveat that we have to account for volume conduction through the skull.
And just to get back to Kim's point that she basically finished on, if you're going to be looking at transcriptomics in different classes of neurons, there must be some significant differences between these neurons which have just local projections that form local circuits, and these neurons that form these really big long axons that go all the way across to the other side of the brain. And that in itself could be very informative, that there are different classes of neurons that may have very different things going on in them.
Mike, one of the most influential papers, I think, is the Antoine paper, having to really think about compensation in these circuits as well. So there may be some aspect of these younger kids where you're having increased brain activity essentially as a compensation and to normalize things, but it doesn't quite work in different age groups, and it may work for certain types of circuits, but very specialized circuits, that compensation can't really be finely tuned. And so we've been trying to understand that in the data as well.
I'd like to add a little bit of a question to all of you about acute versus chronic effects of drugs and what you're seeing in your various preparations, whether they're live humans or live mice or slices in a dish. Would you like to comment about acute versus chronic effects? Because I think we've seen some evidence from some of the clinical trials that there may be some tolerance going on, that it's clear that some drugs work very differently on the first dose versus the 101st dose?
So I'll comment on the human part. So in this NIH Center construct, we only conduct mechanistic single dose probes, which I think is important, but also unfortunate that we don't get into chronic dosing. In the work I mentioned with the BARA 101 GABA A alpha 2/3 modulator, we really see similar responses following 2 weeks of chronic dosing that we see initially. So at least with a 2-week period, we don't see tolerance or changing of electrophysiologic signal. And I think, Mike, and we've talked about this, I think it's going to be different by different drug mechanisms, certainly, but we have to answer those questions. And I think Devin in the in vivo mouse model is set up to do that in a very brisk way and get these early signals, and he can speak more to that.
We're going to try to do exactly what Craig said, and with the caveat that we only can really test for a certain period of time with the MEA in place. However, that could be up to 2 weeks, let's say. So we have the ability to, of course, these mice are being followed, and what we've done thus far is, single dose racemic baclofen versus, daily racemic baclofen for 2 weeks, and then a similar time point of EEG after the last dose. So in other words, a similar time point, but after what you might call chronic administration, chronic being daily for 2 weeks. And we do see in that category of data still an effect at 2 weeks, but different in quantity. So for example, we see the acute dose, affecting gamma and affecting chirp. And at the chronic, it seems to be that it's a little bit less effective. So that's the type of thing where we for each drug should be able to compare that. And whether 2 weeks is really chronic or not, or that's just a time point, we of course have that caveat, but we can compare those types of time points in the mouse to try to get at what Craig is saying. Certainly we can give multiple doses and then look and see what the EEG shows us. I would say just one quick comment. We did a study of mavoglurant, which is unpublished. We don't find an effect of mavoglurant with chronic administration. We wanted to see if we could get target engagement, with an mGluR drug, and whether that's tolerance based upon all the information we've heard from folks in the field, including our colleagues, or not, we don't know yet, because we'd have to compare that to whether it does something acutely. But we could analyze that type of thing to see, to get at your question, Mike. Maybe after 2 weeks, mavoglurant's doing nothing, but if we gave that same dose, which was a dose we took from studies thought to be effective in mice in terms of the preclinical data for mavoglurant, we haven't done it yet, but if we do that as a single dose acutely, are we seeing something? In other words, a reduction in the chronic and with something that works in the acute. So again, we don't have all the data yet, but we want to use this platform for that question.
And just a little follow-up question for you, Devin, about the MEA, the surface electrodes versus the depth electrodes. I know that the depth electrodes are a little bit newer. But it seems like they might actually be from a technical standpoint, easier to use and easier to reuse. Is that true?
Yeah, we don't know yet. We're really just starting these experiments, but we're excited to get some data and really kind of, my plan is to biopsy Kim's brain and Khalil's brain on this. In other words, Kim, of course, knows the laminar specificity of what she's doing in the slice, and we want to see how much laminar specificity we can get. Now, these are 30-channel depth electrodes that basically within about 780 microns are packed into 780 microns. In other words, 780 divided by 32. So you have about 20 to 30 microns or so in between each electrode. So we are going to get high density, penetration of the cortical layers. So we have to optimize, obviously, the technical aspects of the studies, but that's part of what this is funded to do. So I don't have any answers for you yet in terms of technical issues, but we certainly think it's feasible. Certainly, it's just possible to get those data, and I'm happy to share those whenever we have them. I just don't have them yet. But then the idea is cortical physiology, which Khalil is the expert here on, what the single cell recordings and the multi-unit recordings look like in A1, for example. So we'll be able to validate the readings and look at the type of analyses that Khalil has done for many years using his microelectrodes. So that's the idea. That's not data, that's just the idea of what we're going to do. But we want to then see if we get, for example, laminar specific changes in the Fragile X knockout A1 cortex in vivo. How does that correlate with Kim's data? And of course, this question of upstates that she's seen in the slices, is there anything that we can see in the cortex that correlates with those items? So we have these questions that we want to try to address with the depth. But I guess we just don't have the data yet, but that's what we're trying to do.
And one more question, if you don't mind, since we don't have any more from the audience, or do we? Yeah, I think we answered Matt's question about alpha and gamma and arginine. Yep. Okay. Yeah, I just wanted to ask a general question about scalability of these methods. And in particular, it takes 10 minutes to get one of these high density EEGs, and it generates so much data that it's really a lot of work to analyze it. And I know, Ernie, you've done a lot of work on the IT side to just enhance the speed and the uniformity of processing of this data. But when we're starting to look at large-scale clinical trials that might want to use these methods as outcome measures or methods for stratification at the very least, can you say a little bit about what it would be like to have 200 subjects in a clinical trial using these methods?
Yeah, so I think a lot of our work has been, first of all, trying to blind the data so it's sort of it's a non-biased evaluation of it, and we've really worked on the software side on developing a pretty strong pipeline that can, I think our record is maybe between 150 to 200 EEGs in a weekend, and so we're at the point now where we can really do some good semi-autonomous processing. The analysis is all scripted in a way that we can get nice outputs and graphs right from the end, and so I think we're in a good stage for that. We certainly have a lot of experts who are sort of validating the work, giving us input from their cutting edge pipelines. And so I think the field is there now, and certainly the computational power. Like I remember 5 years ago when we started, it would take about 3 hours per subject just to get our artifact processing. We've now simplified that down to 1.5 to 3 minutes a subject. And so there's been some great advances there as well.
Really important. We have another question.
It's a comment.
Oh.
I think to add to that, I think our limitation is more going to be clinical trial sites and getting the EEG technology, getting the nets on the heads, and all the behavioral and other support that goes into doing that work. And I think as a network through NeuroNext, funded by NIH, Liz Berry-Kravis has been, leading about 20 sites to do some of this work in younger children. But I think that's going to be the bigger issue. I mean, I sort of feel like what Ernie's doing in the analytics and with Devin, it's kind of like computer chips. The technology just gets better really quickly in the analytic power. I think more of the human element of getting the signal, getting the nets on, doing that with fidelity is what we need to work the most on to get the use widespread.
Okay, are there any more questions? We've got some good comments in here for folks to read, but any more questions for our speakers? Well, we're perfectly on schedule. Huh. Okay, where do you hold the clinical trials for young adults? And how can you apply to get involved? A parent question. That's what all the parents want to know.
Well, we're always doing clinical trials at Cincinnati Children's Hospital. I think that COVID has been a challenge on trials. Our trials have never stopped, but they've certainly slowed. The trial work in this center is going to take many months longer working with the NIH on some specifics of design. So I don't think specific to the next iteration of this center will the clinical trial work happen for maybe another 6 months or so. But anyone can contact myself, Cincinnati Children's Hospital. We're always happy to interface with families about opportunities. And honestly, if you're on the FRAXA listserv, you'll get an email about every trial enrolling anywhere at any time. So you can have confidence that what's out there and what's exciting and available.
We're always recruiting for some trial or another. Well, guys, that was fantastic. I just want to thank all of our speakers for amazing presentations and really exciting technologies that they're developing. We're right on schedule. And so I guess that concludes our seminar. Thank you very much, and everybody have a nice day.
Thank you,
Thanks,
everybody.
Mike. Thanks, Katie.
Yeah.
Thank you.
Okay, thanks, Eric.
Thank you so much. Thank you.
Thanks, everyone.